Josh.
All right, I think we're in business. Just wanted to start today with a couple of announcements of sorts. I've been asked to let you all know about the Carnival of Caring. I talked with one of the organizers yesterday, and I said, wait a minute here, you're giving people free food? I thought this was a fundraiser sort of thing. How's this going to work? You give them food? I thought you wanted to make money out of this. But the idea apparently is they want to prime the pump. They want to bribe you to come to this thing by giving you free hot dogs, chips, and soft drinks, and then have you play games while you're there and charge you like $1 a game or something like that. I think the games that they're going to be playing are the same games that they do for the Transplant Games of America. And it's the Ohio team, which is apparently well represented by Wright State, that the money is going to help. So anyway, if you have a chance, if you're looking for a place to get a free lunch tomorrow, and that is tomorrow, go to the Student Union Atrium, and I'll take care of you by getting you a free lunch that way. How about that? You don't have to buy the ticket to the game. Just take their hot dogs and leave, and we'll all be in good shape. I said a couple of announcements. The other one is this one. I'm going to show you this one again on Thursday at the start of class, maybe have it in the background while I start talking with you about things. But you guys all know and love the freshman lab, right? You guys spend some time there every week. The people who really make that lab run are students, typically undergraduate students. You guys get to know the graduate TAs pretty well, who grade your reports and quizzes and things like that. But behind the scenes, and sometimes not that much behind the scenes, that are undergraduates. They're looking for a few good undergraduates to work in there starting in the spring semester. Marcy Wendelin is the director of the freshman biolabs. She has had an amazing track record of being able to recruit in really good students to work in that freshman prep lab during the past 15 or so years that I've known about what goes on in there. So if you like hanging out in that lab, if you'd like to be more involved with the lab, if you'd like to get paid for helping out with the labs and see what goes on behind the scenes, how many crayfish die during the course of the experiments or whatever, because that's what you're doing in lab this week, by the way. There will be a lot of crayfish involved. If you want to be involved with that sort of thing, that's a neat opportunity. If I was looking for a job as an undergraduate, I'd be all over this. So anyway, there's some information for you there about this employment opportunity, and it will be something that I'll show you again at the beginning of lecture on Thursday. But we got serious biology work to do. Today, I want to talk with you about half of metabolism, right? We'll just get the front half of it pretty much out of the way. Today, I want to talk with you about catabolism. Maybe it's better to refer to catabolism as the back half. You guys will decide in the course of the next week if this is the front half or the back half. But today, I want to talk with you about the central part of metabolism that's involved with extracting energy from glucose. That's what's on the agenda for today. It turns out that it's a sufficiently complicated set of ideas that I want to convey that I'm going to spread it out over both today's lecture and Thursday's lecture, and it'll be a week from today when I'll talk with you about the other side of the coin, the yin to this yang, which is essentially photosynthesis, the process by which organisms capture energy, photons associated with light, and convert that energy into covalent bonds that have energy stored within them, particularly those bonds that are part of glucose molecules. Right? So that'll happen a week from today. We'll talk about photosynthesis, but today I'll talk with you about how it is that we extract energy from those bonds in glucose, and then we'll get into even more detail about that on Thursday of this week. So that's what's in your short-term future for what I'll be talking with you about. As a little bit of a warning, let me tell you this. This is actually a pretty easy lecture for me to give. I don't have to have a lot of information in my head. And so it takes me like 5 minutes to refresh my memory from looking at the lecture notes. But that makes it actually one of the more difficult ones, content-wise, for you guys to wrestle with. What I'm going to try to do with you today is gently teach you and expose you to some rules that will let you make sense of all the rest of this stuff. And there's a couple of different kinds of rules. I'm going to be talking with you about a really centrally important metabolic pathway, glycolysis, taking glucose and breaking it down into 3 carbon molecules, a 6 carbon molecule into 2 3 carbon molecules. So I'm going to talk with you about glycolysis. And there's a couple kinds of rules that I'm going to try to draw your attention to. One is just the chemistry of it, how you could recognize that we're going from an energy-rich molecule to ultimately some things that are lower in energy, and how it is that that process needs to play out in a cell. So I'm going to talk with you about the chemical rules. But I also want to talk with you about the nomenclature rules. And I'm particularly interested in making sure you're comfortable to some extent with the names of the enzymes that are involved with catalyzing those reactions. There's a couple ways that this could happen. You guys could invest a couple hours and commit to memory the names of all the enzymes that I'll be talking with you about today and Thursday. It's a pain. I've done it many times, and I think that might be the most important point that I can make to you here. I've done it many times. I don't think any of us memorize in such a way that you will remember it indefinitely. Things that you memorize tend to fade away over time. And I've had to memorize the names of these enzymes probably a half dozen, maybe a dozen times over the course of my professional career. You could do that. It's going to cost you a couple hours. I think it'll serve you well for the next test coming up at the beginning of November. But I bet you by the time the final exam rolls around, you've forgotten most of those names and you'll have to memorize them again. My preferred approach is just getting you to be comfortable with the nomenclature, how it is the names are derived. There are some pretty simple rules so that I can give you a scenario, and you could either recall the name of the enzyme from memory, or you could figure it out just from what's involved with the process. Right? That's my preferred way of understanding these things. And so again, this is easy for me to do because I know those basic rules. I'm going to try and convey the basic rules to you, but it requires you guys to do some work then to make sure you know how to apply those rules. And I think that means you got to spend some time with either the PowerPoint slides that are available on Pilot or the pictures straight out of your textbook, just to really get comfortable with what's going on with these pathways. Okay? So, with that said, let's get working on talking about catabolism. And there's this one concept right at the beginning, this idea of redox reactions. And I'll be honest with you, I try to be honest with you all the time, but let me be really open with you about my honesty here. I'm a little insecure about redox reactions. I've wrestled with this idea since I was an undergraduate, taking chemistry classes and all the way through. And so I'm not going to stand up here and tell you, oh, redox reactions, this is trivial, it's silly, it's easy stuff. You know, but the basic idea is pretty solid. Here's the thing. You guys know that bonds that involve interactions between atoms with similar or the same electronegativity are the bonds where you store a lot of energy. Okay? Covalent bonds, non-polar covalent bonds, those are the ones where the energy is at. And conceptually, the way to make sense of that is this, is that I like to think, and I think it's actually based in the reality, when you've got a covalent bond between a hydrogen and a carbon, the electron, or the electrons that are being shared between those two atoms, are often far away from the nucleus of either the hydrogen or the carbon. You could think of them as if they were high altitude, right? They were far removed. They have a lot further to go to fall to get close to the nucleus of either the hydrogen or the carbon. To get far away, to have that high altitude, they have to have a lot of energy associated with them. And as they fall, and as they get close to atomic nuclei, they have less energy associated with them. And that energy has to go somewhere. Which law of thermodynamics is that? That's the first law of thermodynamics. Energy can neither be created nor destroyed. So if you have a high energy electron and it falls to a lower energy state, that energy has to go somewhere. All right? So when we're talking about energy in covalent bonds, we're often talking about going from a carbon to hydrogen bond to maybe a hydrogen to oxygen bond. That's pretty obvious, right? When you go from nonpolar covalent bonds to polar covalent bonds, those molecules, I think you guys know now how to look at and say, wait, this is an energy-rich molecule, that's an energy-poor molecule, all right? What redox reactions are all about, it encompasses that, but it takes it a little bit further, and it's subtle in the sense that redox reactions are tracking where the electrons go, and redox reactions are mindful of the fact that if the electrons are in the same kind of bond, but at a lower energy state, you've still had what's effectively a transfer of energy. The electrons moved, when they moved from high energy state to low energy state, there's still some energy that has to be transferred. So that's what's at the core of this redox business. So let me put it to you simply, all right? I think you don't need me to tell you this. Glucose, at least at this point in the class, glucose is an energy-rich molecule, right? And carbon dioxide is an energy-poor molecule. Picture the chemical structures. I'll show you a picture in a few minutes, right? Glucose, lots of high energy bonds. Carbon dioxide, low energy bonds. In lecture on Thursday, I went way out of my way to talk with you about taking a match and putting it next to glucose and setting the glucose on fire, combustion. What you're doing is you're combining glucose with oxygen to give rise to products. Those are the substrates, the reactants. Those give rise to the product of carbon dioxide gas and water. Higher energy molecules end up low energy molecules. Energy gets released along the way. Glucose is high energy. Carbon dioxide and water are low energy. What you do when you burn glucose is you oxidize glucose. And when we talk about oxidation, when we say glucose has been oxidized, what we're really alluding to there is you've taken electrons that were in high energy states and you've transferred them ultimately to molecules in a low energy state. When that happens, you say that something has been oxidized. Okay? The flip side of oxidation is reduction. So when you take a low energy electron and you make it into a higher energy state, that's called reduction. And that together then are what we mean by these redox reactions. Whenever you're doing the one, whenever you take an electron, whenever you oxidize something, use it that way, whenever you oxidize something, you have to simultaneously be reducing something else. The two are part and parcel. You can't do one without the other. And so here's the bottom line. Everything that I'm going to be talking with you about today and on Thursday, and on next Tuesday for that matter, are all redox reactions. Bear in mind, keep in mind that we're talking here about high energy electrons becoming lower energy electrons, and keeping track of what's being oxidized and what's being reduced. Right? Those are the players. That's the scorecard that lets you see the names of the players that are involved in here. Everything involved with photosynthesis and glycolysis is a redox reaction. Okay? And so try to keep your eye on those balls, and that'll help you make more sense of what's going on. So let me show you some pictures that might make a little bit more sense. This is more of a general kind of picture before I start talking with you specifically about redoxes. I showed you this on Thursday to give you this sense that, you know, combining hydrogen gas with oxygen gas, two very energy-rich molecules, right? Hydrogen gas, covalent bond between two things with the same electronegativity. By definition, hydrogen has the same electronegativity as hydrogen. And oxygen, the same scenario. When you combine hydrogen gas and oxygen gas, you can easily make water. The energetics are very favorable. The delta G is very negative. And you can do that in an explosive way that releases an awful lot of heat and some light, right? That explosion, though, is not very practical for living things to do. Right? You don't want to have lots of hydrogen gas exploding inside of all of your cells all the time. It's going to be uncomfortable at the very least, and probably, you know, just plain bad idea. What we do instead in living things is we facilitate that same chemical reaction, going from hydrogen gas, in a sense, to water by combining it with oxygen, but we do it in a gradual, stepwise fashion. You could think of it as a bucket brigade, right? You got water at the top of a hill, and it's pretty easy to transfer it down to the bottom of the hill, but we do it in a controlled fashion. Instead of just pouring it and letting it spill down in a little rivulet down the hillside, we take a bucket, and we have it handed off to another bucket. It's handed off to another bucket further down the hillside, and along the way, energy is going to be captured through the generation of ATP. So you'll see that time and again when we're talking about glycolysis and photosynthesis, is that we're always going to be talking about energetically very favorable reactions, very negative delta Gs, but rather than do it in one big flash, we do it in a gradual, stepwise fashion. Okay. So here's what I mean by redox reactions. Here's some pictures that might make that a little clearer. What I said a few minutes ago, a little bit clearer. Sodium and chloride. It's a match made in heaven. Nature loves to facilitate the interaction between sodium and chloride. You guys will remember, I'm sure, that sodium's got that one lonely electron in its outermost shell. Right? And again, nature really likes things to have either completely empty or completely full outermost shells. For sodium to get a completely full outermost shell, it needs to be able to coax away 7 electrons from something else. Or, if it wants a completely empty outermost shell, it just surrenders one of its electrons, gives it, dumps it off, pawns it off onto something else. Neither of those are great from a nature perspective. One of them requires a 7 negative charge associated with the sodium. That's pretty horrific, right, from nature's perspective, that really local, intense negative charge. The flip side is a local positive charge, because you've surrendered one of the electrons and now you've got an extra proton in the nucleus relative to the electrons that are out in the orbitals around it. So given the choice of the two, -7 or +1, it's pretty easy for nature to say, gee, I really like it when the outermost shell is completely empty or completely full. That's my first priority. My second priority is let's not go crazy with the concentration of charges. So 7 is insane. That's really crazy. 1 is pretty crazy, but it's not as crazy as 7. So sodium just happily gives away electrons, an electron, whenever it has the chance. Chlorine, in contrast, it's the exact opposite. Chlorine needs just 1 more electron to round out its outermost shell. It can pawn off 7 electrons, or it can pull in 1 electron. It's a lot easier to pull in 1 than to get rid of 7. And what ends up happening, we're talking here, I keep saying the word electron. We're talking here about a transfer of an electron. The electron is going to go from sodium to chlorine. Chlorine acts, well, let me put it this way. Sodium gets oxidized. Chlorine is a reducing agent and ends up taking up that electron. Okay, so this is a redox reaction. And this is a pretty easy one to follow because the charges are showing you where the electrons have moved. Sodium gets oxidized, chlorine gets reduced. It's a redox reaction. So this one's pretty obvious. The next one is the one that's it's more germane to what we're talking about for biological systems. Let's talk about what happens during the course of the combustion of methane. Okay, so here, what we're going to do is we've got our methane gas. We're going to combine it with oxygen gas. Those are the reactants or the substrates. The products are going to be carbon dioxide gas and water, and there's also going to be some energy that gets released along the way. So what happens here? We've got a lot of energy-rich electrons associated with the methane. We've got energy-poor electrons here and here, energy-rich electrons here. So everything here is energy-rich electrons in those covalent bonds. Excuse me. And over here, we're talking about covalent bonds that are polar and don't have a lot of energy. The previous image let you see how the electrons were moving because the charges were changing. Right? It was obvious that an electron was moving. Electrons are moving here too, but here the electron motion isn't so much something that you track by looking to see where the charges have moved, but rather where the energy state of the electrons have changed. Here you've got high energy electrons. If you like to think of them as high altitude electrons, I can see how that would help. Here you've got low energy electrons. They're closer to the nucleus. They're lower altitude electrons. In a scenario like this, the high energy electrons are going to get oxidized, conveniently here because of their interaction with oxygen, and we're also going to be reducing these electrons as they end up becoming associated with water. And we have, again, just as before, a redox reaction. Keep your eye on the ball. The ball here is we've got high energy electrons, and we've moved them into low energy statuses. That's a redox reaction, just as it was with the sodium chloride, just as it is with all of these other reactions we'll be talking about for glycolysis and photosynthesis. Yeah.
I noticed on the diagram on the methane and the oxygen gas, the little dot structures they have for the electron bond are pretty much in the middle between that when you get carbon dioxide, they're all and water, they're all gravitating towards the oxygen.
Mm-hmm.
Right.
So he's drawing attention to the fact that just as I'm talking about these electrons being far removed from the atomic nuclei, they've done a nice job in the cartoon rendering with these Lewis dot structures. Notice how here, this is a high energy covalent bond. The electrons, those dots, are shown equidistant between the carbon and the hydrogen. That's to help you recognize this is a high energy bond. Compared to when you look over here, the dots are always close to the oxygen as opposed to the hydrogen or the carbon. Here the dots are much closer. They're not equidistant between the hydrogen and the oxygen. They're a lot closer to the oxygen. And so that is deliberate, and it's playing into this idea that I'm trying to conjure of high altitude, far removed from the nucleus. Right? So these reduced things have electrons that are low energy. They're close to the oxygens. Here, these things that are going to get oxidized have high energy electrons. All right? Bottom line, a redox reaction. What's going on is we're tracking, we're keeping our eye on where it is electrons are moving. There's a shell game of sorts that's going on. You need to be able to keep your focus on where the high energy electrons have gone. They started here, there's a lot of shuffling that happens, they end up over there, right? Something gets oxidized, something gets reduced. The things that are in play here are where these high energy electrons are moving through the process. One last thing. with respect to these redox reactions. Today, I'm going to talk with you for the rest of the lecture about specifically how it is that glucose is involved in a redox reaction. It gets oxidized to become carbon dioxide gas. The oxygen gas gets reduced to become water. And along the way, of course, we're releasing some energy. So let's get to talking about glycolysis. Before I get to talking about glycolysis, let me just first pause and say a little bit to you about this idea about aerobic versus anaerobic respiration. I think you guys already know what aerobic and anaerobic means. Aerobic means in the presence of oxygen. Anaerobic, an prefix means without, right? So anaerobic is absent oxygen. Aerobic is with the presence of oxygen. For those of you who exercise, even those of you who don't, you've probably encountered this idea before. Aerobic exercise is the exercise where you're working, but you're not really short of breath, right? You're continuing to breathe the whole time. Anaerobic exercise is where you're working harder than you can bring oxygen to your muscles to keep things going. Right? So weightlifting, intense bursts of energy that go out, that's typically considered anaerobic exercise because you're doing it without the benefit of all the oxygen that you might have been able to benefit from because it's such an intense burst. Aerobic exercise is something more like swimming or cross-country running, where there you're expending energy, but you're doing it by fueling the flames by bringing in oxygen all the time. Living things, I just gave you some examples of us as living things, vacillate between aerobic and anaerobic all the time. Why? What's the deal? It turns out that when you're burning glucose, you get a lot more energy out of glucose if you can combine it with something like oxygen than if oxygen's not available. So for aerobic respiration, when we're talking about taking energy out of glucose in the presence of oxygen, when we've got everything working just right, we as human beings, but E. coli as a bacteria, we can get on the order of 32 to 34 ATPs per glucose. Okay, that's what happens during aerobic respiration. That's as good as it gets. For anaerobic respiration, glucose is an energy-rich molecule, right? But the end point of water isn't going to be something that we can create, which is a very energy-poor molecule in terms of its bonds. We can only get it part of the way toward carbon dioxide and water. You only get 2 ATPs for anaerobic respiration, whether you're a human being or whether you're a prokaryotic organism. So, if you have the choice as an organism to extract energy from glucose in the presence of oxygen or the absence of oxygen, it would be really a bad choice to decide to go anaerobic. You get how much more? 32 more or less extra ATPs if you can use oxygen to help you with this process. The absence of oxygen, you get what you can get, and that's 2 ATPs. So, we have to sometimes work without oxygen, and the 2 ATP we end up finding ourselves being thankful for, but when you have the chance to use oxygen, we will burn glucose in an aerobic fashion. Bottom line, aerobic, you get much more ATP than when you're doing this anaerobically. Along the way, I'm going to be drawing your attention to where it is you specifically need to have oxygen to play these games that we'll be talking about further down in this lecture outline. All of this stuff with glycolysis can be done anaerobically. It's only when you start getting down to the electron transport chain that we need to start talking about needing oxygen present to get every last little bit of energy out of our glucose. So the beginning steps can happen in the absence of oxygen, but if you want to get all the energy you can, you need oxygen to help you drive the electron transport chain. Okay, glycolysis. Let me tell you what you guys are in for as I start to talk with you about glycolysis. Glycolysis is, I say in the lecture notes, it's a tale of 10 enzymes, right? It's Dickens, right? Tale of Two Cities, right? Here we got 10 enzymes. Whole much more interesting story than the Tale of Two Cities. There are 10 enzymes that are catalyzing this process. Again, I think you guys should be starting to be pretty comfortable with this. It's a metabolic pathway. You take a molecule at the beginning and you take it in a stepwise fashion until you end up with some other molecules at the end of the process. In a nutshell, in a word, what we're doing is we're taking glucose and we're converting it in a stepwise process to pyruvate. Okay, that's what's going to be happening during the course of glycolysis. And of course, glucose is primarily there as an energy storage molecule. We're using glucose to store energy, and so this process shouldn't surprise you to hear that along the way, at the end of this process, we will have released some energy in a form that's usable to cells, and in simple English, that means we'll make some ATP, right? So this 10-step process going from glucose, a 6-carbon molecule, to 2 3-carbon molecules, pyruvate, that process of glycolysis results in the creation of some useful energy to cells in the form of ATP. Specifically, 2. We can make 2 ATPs simply by driving that process of converting glucose to pyruvate. Remember, this is a bad deal energetically, in some sense. Glucose, 686 kcal per mole. Pyruvate has a pretty high amount of energy associated with it, but it's not useful. You can't put pyruvate into a vending machine. Okay? ATP you could put into a vending machine. It's the dollar bill of the energy currency. That's only 7.3 kcals per mole. Okay? We got 2 of them, so that's going to the bank with your $100 bill and them giving you $2 in exchange, literally 2 $1 bills. It's a bad deal, but if you want a Pepsi really badly and you only got vending machines to work with, it's a deal that you're going to take. There's no oxygen involved. This can happen anaerobically, and you can get some useful energy. There's only so much you can do with glucose. There's stuff you can do with ATP. If you really want the ATP, sometimes you're going to have to be prepared to take this really bad exchange rate and get your 2 ATP per glucose. So this is the first step in the process. Again, it's a 10-step process. That's the first step. But it creates for us 2 ATPs in those pyruvate molecules. And the pyruvate molecules are things that we can then submit to another pathway. Come next, the citric acid cycle, and we can then crank that cycle and get more ATPs out of it. Okay? This is an ancient metabolic pathway. There's some metabolic pathways that people who are trained in such things can look at and say, oh, this is relatively new under the sun. This hasn't been around for too long. Glycolysis isn't one of those things. Glycolysis is smack dab in the middle of metabolism. All the rest of metabolism seems to be tacked on in some way, sort of kludged on onto glycolysis. The stuff that, so glycolysis was probably one of the first things that living things on this planet had to work out. Let me just tell you a little bit about what it is I mean. Say that you eat something with, some sugars in it, right, that aren't glucose. How do you get energy out of sugars that aren't glucose? Because sugars have energy in them. How do you get energy out of a molecule? How do you get energy out of a molecule a sugar molecule that's not glucose? I got an easy pat answer to that question. You make it into glucose. Okay? Anything that's not glucose, if you want to get energy out of it, and it's a sugar, you convert that thing into glucose. Why? So you can run it through glycolysis. There is no equivalent pathway for any other sugar. Everything else gets channeled into glycolysis. What if it costs you an ATP or two to channel that thing into glycolysis? You do it. And at the end of glycolysis, you may end up with no net gain in terms of ATPs, but you do it because you're making these other valuable molecules like pyruvate and also these NADHs, which you might be able to get some energy out of if you can do aerobic respiration. Okay? So that's part of what I mean. This is a really central pathway. Everything gets channeled or funneled into glycolysis. And then let's say you need to make stuff like an amino acid. You know how you do that? All of the amino acids get shunted out of either glycolysis or the citric acid cycle. Right? So when living things found that they needed to make these different amino acids, the way that they ended up settling on how to make them is, I guess we're going to take some of them out of this pathway and use them for this alternative purpose. Again, that happens either for glycolysis or the citric acid cycle. It is a central metabolic pathway. And it actually works the other way around, too. Say you eat a lot of alanine, an amino acid, and you want to get energy out of it, you know what you do? You convert it into something that you can plug into the glycolytic pathway. So again, bottom line, this is an ancient pathway. It is central to metabolism. And it's something that all living things share in all of their details. The enzymes involved are the same whether you're talking about an archaebacteria, a bacteria, or a eukaryotic organism. This is the bottom line. Go from glucose to pyruvate, ATP, and NADH. You do it through this process called glycolysis. It's a 10-step process. What's going to happen to you now is I'm going to expose you step by step, again, to those 10 enzymes that do the business of glycolysis. So let's get started. That's a high-level view. That's what, 50,000 feet, glucose to pyruvate. Now we're zooming in, right? Now we're at like 20,000 feet. We've broken it up into a 3-step process, all right? Pretty soon, these steps are going to get broken down further so that you end up with a total of 10. But the general strategy for extracting energy from glucose that all living things use, doing glycolysis, the general strategy is this. It begins with an investment phase. It may seem a little strange. You've got an energy-rich molecule, glucose. The way you get the ball rolling is you put more energy into it still. So there's this energy investment phase where 2 ATPs get expended for every glucose that you want to metabolize. Okay? Convert 2 ATPs to 2 ADPs and inorganic phosphates. And after that energy investment phase, the next to follow is the energy payoff phase, shown in darker blue there, where here you take 4 ADPs and 4 inorganic phosphates and create 4 ATPs from them. And also as part of that energy payoff phase, we do these redox reactions where you convert something called NAD and some electrons and hydrogen ions, to NADH and hydrogen ions. At the end of the process, we've made 2 pyruvates and 2 waters, as well as these NADHs, the 4 ATPs, and the lost 2 ATPs for a net gain, 4 minus 2, of 2 ATPs. I'll steal some of my thunder for the end of the lecture and the lecture on Thursday and tell you the NADHs have a lot of energy associated with them. Those electrons that sort of get carried over with the H, get used to make a lot of ATPs. But that only happens with the electron transport chain. That only happens with the presence of oxygen. Okay, so you can only do that aerobically. If you're talking about anaerobic respiration, we can just more or less forget about what's going on here. Nothing's going to happen. All that you're going to get are the net gain of 2 ATPs from this energy payoff phase. Okay? So again, I keep warning you here. You guys are probably really afraid of what's going to happen. It's not that scary. Let's talk about the enzymatic processes that are involved to take you through these steps. This comes from your textbook. They do have a neat coloring scheme here, and it's going to be applying all the way through the rest of what we do. Glycolysis is this. Ooh, it's nice that they tell me what that color is. That's teal. These pink colors are salmon. That's when we're talking about what happens with the citric acid cycle. And then oxidative phosphorylation. Oxygen, again, is what happens in the electron transport chain, and it's got that purplish color. Where these different reactions take place is different. Glycolysis happens exclusively in eukaryotic organisms within the cytoplasm. The citric acid cycle happens within eukaryotic organisms exclusively in the mitochondria. And the electron transport chain also happens exclusively in the mitochondria. So we've talked about these intramembrane-bound compartments. The mitochondria is sort of the dynamo, the powerhouse of the cell. The cytoplasm is where glycolysis occurs. The enzymes that catalyze glycolysis, the conversion of glucose to pyruvate, reside exclusively in the cytoplasm. The enzymes that do the citric acid cycle hang out in eukaryotes exclusively in the mitochondria. And the enzymes that are involved with extracting energy from NADH, right, the electron transport chain, reside exclusively in the mitochondria as well. For prokaryotic organisms that have no internal membrane-bound compartments, all that has to happen in the same place, the cytoplasm. But for you and I, we have the luxury of being able to use a specialist with the citric acid cycle, and even better, the electron transport chain. The structure of the mitochondria, you will see, sets things up in such a way that the electron transport chain can be run very efficiently by mitochondria, more efficiently than it could be run and is run inside of prokaryotes. So again, this compartment of the mitochondria turns out to play a really important functional role because of its structure. But again, we're going to start by talking about glycolysis. Let's see how it is we convert glucose to pyruvate. There's glycolysis again. It's a multi-step process. Remember, at the start of the process, there is an energy investment phase. You see that structure in the upper left there? That's glucose. You've seen it a bunch of times before in this class. Here's some salient features, some things that I hope pop out at you about glucose. It's got 6 carbons. It's got 6 carbons, all right? One of the things that might help you as we go through this is keep track of the number of carbons in the molecules that are involved. At the start, it's going to seem trivial because it's going to be a 6-carbon molecule for a long time. It's going to become 2 3-carbon molecules, but the numbers need to add up to 6 all the time. 2 times 3 is 6. When we get to the citric acid cycle, keeping track of the number of carbons is going to get a little bit trickier, and you'll see, but it still always has to add up, and you always have to bear in mind you started with a 6-carbon molecule. So glucose, one of the salient features, it's a 6-carbon molecule. Another salient feature, if you just went through this structure and counted the number of high-energy bonds, you would be able to say, oh, that's a pretty high-energy molecule, right? That's a high-energy bond, that's a high-energy bond. All around here, there are high-energy bonds. And remember also that at the start of glycolysis, we kind of primed the pump. We invest some energy to really juice this molecule up. It was 686 kcal per mole at the start. Well, think how much more energy it gets when you tack on a phosphorus, a phosphate group, to the end of that molecule. All right? If you have the choice, if I asked you which of these has more energy, this molecule or this molecule, everything about this is the same as this except for that phosphate group. You should just accept whenever you see something like that, if there's a phosphate group on it, there's energy there. That's a higher energy molecule. And it makes sense because, again, we're investing energy here. This is a process where you've got two substrates or two reactants, again, equivalent terms. You have glucose and ATP, and you have two products of the reaction. Chemists will show a chemical reaction as having an arrow. You go from the substrates to the products. The products here are ADP and glucose 6-phosphate. So tacked that phosphate group on. The arrow tells you there's a chemical reaction. This is the direction in which the reaction tends to play out. This is where it's most energetically favorable. It tends to be going from left to right because of that arrowhead. And the way that chemists will do this is if it's a catalyzed reaction, they'll give you the name of the catalyst over the arrow. Here under the arrow is they put the other arrows on top. But written by the arrow will be the name of the catalyst that catalyzes the reaction. All of these reactions we're going to be talking about for glycolysis are catalyzed by biological catalysts, specifically by enzymes, and each of those enzymes is going to have a name. The first of those enzymes' name is hexokinase. All right? Now you could commit that to memory. I got no beef with you if you decide you want to memorize that the first enzyme involved in glycolysis is hexokinase. Or you can kind of wrap your head around the way that these enzymes got named. There's a method to the madness. There are 3 classes of enzymes that you're going to encounter. This is the first instance. It will be an example of one of those classes. One of those classes are kinases. For the rest of your biology careers, whenever you hear the word kinase, it's always going to mean the same thing, unless you're in some sort of bizarro world where by agreement you decide to use kinase to mean something else. For normal people, kinase means the enzyme involved is transferring a phosphate group from something to something else. That's all that kinase means. It's a $64 word to say it's an enzyme that catalyzes the transfer of a phosphate group from one thing to another thing. There are a lot of different kinds of kinases. All kinases transfer phosphate groups from one thing to something else. What is it that hexokinase is doing? Its primary job, what the reaction involved here is it's taking a phosphate from ATP and it's transferring it to something else, to glucose, right? Great example of a kinase, right? What is the molecule that this kinase is acting upon? Glucose, right? How do you suppose they came up with the name hexokinase? I'll tell you, if they wanted to be, cool and, friendly toward people who wanted to wrap their head around this stuff, I think they would have called it glucose kinase. They were not so lucky. The guys who named hexokinase decided to go with something other than the obvious glucose. They went with hexokinase. Hexo is a reference to the substrate for the reaction. Okay? Hex means 6, right? And so hexo, the O is like a tip of the hat to the sugar, the O's of sugar. So you have hex, a 6-carbon sugar is being kinased. Smush it all together and you got hexokinase. So, what do you guys need to know about this? I'm hoping you guys will walk away with this sort of like a scorecard such that now you'll remember kinases mean that a phosphate group's getting transferred to something else. Okay? That would be cool. If you look at some of the old tests, you'll see I ask questions like this. I'll say in the first step of glycolysis, an enzyme takes a phosphate group off of ATP and attaches it to a 6-carbon molecule named glucose. What's the name of that enzyme? I'll give you 5 choices. Okay? And just let that sink in. What do you think? What kind of choices might I give you? I'll tell you this. If kinase isn't part of one of your choices, it's a loser. Right? First and foremost, what's going on here is we're transferring a phosphate group from one thing to another thing. Only kinases do that. Not dehydrogenases, not isomerases, not any other kind of enzyme name. Kinase had better be part of the answer. Okay? And for this one, I doubt that I would give you any choice other than 1 that had kinase and 4 that didn't have kinase. So I'd be pinging you on that. It's possible, if I'm feeling you guys, if I need to bring down the class's average score on the test, if I feel like, boy, you guys are doing too well, I got to bring you down to earth, then I might give you the choice of a couple of different kinases. The kinase you want to go for here is the one that's working with a 6-carbon molecule. Maybe the hex thing will come to mind, and you'll be able to recognize it as the kinase that's involved here. Okay? So that's how you start things off. Remember, it's an energy investment step. Okay? The phosphate group came off ATP, and we've made ADP. So in some sense, you could argue we've lost energy. We've transferred it from something that's pretty useful to the cell, an ATP, to something that still needs a lot of work. Now, glucose 6-phosphate. In terms of the name of the substrate, you guys, I know, are comfortable with talking about glucose, right? That name by itself, well, I guess that could be intimidating if you told somebody at a bar, yeah, in class today, we talked a lot about glucose 6-phosphate. You know, you want to practice it before you do that. You don't want to stumble over it. You know, it has to roll off the tongue. Glucose 6-phosphate, like it's no big deal, right? There's nothing scary about that name, right? You can use it to intimidate and impress people, but, you know, well, look at it. It's glucose, and that means it's got a phosphate group stuck onto it somewhere. Where's the phosphate group stuck on? How about onto the 6th carbon in the circle? Going clockwise around the circle, 1, 2, 3, 4, 5, 6. Where's that phosphate group? It's attached to the 6th carbon, and that's the way they always do this nomenclature. You start clockwise at noon, and you work around, and you do your counting. 6 is where that phosphate group is attached. So there's no great mystery or mumbo-jumbo associated with the name. It's following some pretty simple rules. That compound is glucose 6-phosphate. Okay? What happens next? Well, I told you at the start, we have an energy investment phase. What do you suppose has to happen next? We're done with the energy investment. Halfway done with the energy investment. And now what we need to do is we need to rearrange the structure of the glucose a little bit so that it's ready for the next step in the process. Okay, so glucose is fine. We dressed it up. We invested a little bit of energy to give it that extra phosphate. But now we need to just tweak the structure a little bit so that it's ready for the next enzyme in this bucket brigade, in this change. In a word, what we need to do is make an isomer of glucose 6-phosphate. Okay? Same chemical formula, different chemical structure. That's what an isomer is. And you know what? We got a second class of enzymes to talk about now. Enzymes that make isomers Right? Go from one isomer to another isomer, same chemical formula, different chemical structure, cleverly named isomerases. Kinases are enzymes that catalyze the transfer of phosphate groups. Isomerases make things into other isomers of that thing. Right? So we've got an isomerase at work here. This isomerase is named phosphoglucose-isomerase. If you think about it, it's a pretty clever name. Let's focus on the gluco. That's glucose, right? It's glucose with just one phosphate on it, so phosphoglucose-isomerase. And what happens here is glucose 6-phosphate becomes fructose 6-phosphate. Same chemical formula, little bit of a different chemical structure. What we're doing is we're poking this carbon up and bringing this carbon up with a bond to that oxygen. So a little tweak to the structure of glucose 6-phosphate to give us fructose 6-phosphate. What do you guys need to know? I'll tell you what you need to know. I want to make sure you guys know what isomerases are. Don't confuse isomerases with kinases. I don't think that's asking very much, frankly. It's a little bit of vocabulary, but that's one of the main things I hope you take away from this. And I hope you'll recognize, yeah, that's an isomer of that. Oh, that name makes some sense, right? You could memorize that name, or you could simply say, yeah, I understand that name. I'm not intimidated by the name. If I gave you this structure and this structure, and I asked you which one of those is fructose 6-phosphate and which one is glucose, come on, how hard is that going to be for you to work your way through? All right? That's pretty obvious. If I gave you this structure and I asked which one is glucose 6-phosphate and which one is fructose 6-phosphate, I think most of you, it'd be pretty reasonable to expect that you'd be able to discern between fructose versus glucose, because you've seen glucose so many times now. But that's kind of pushing it. I don't know that I want to go that far. I cannot, in a class like this, ask you to draw for me the structure of fructose 6-phosphate. That might help you wrap your head around the level or the way in which you want to learn what we're talking about here. You know, above it all, I think the most important thing is I don't want anybody in this class to get intimidated by the mumbo-jumbo, jargony kind of chemical names that chemists have given these structures or that you get from these enzyme names. Fructose 6-phosphate shouldn't intimidate you. It's not that big a deal. That's what fructose looks like. There's a phosphate group stuck on it. Where is it? It's on the 6th. Okay, I guess we should call that fructose 6-phosphate. Anybody want to come up with another name for fructose 6-phosphate? It seems like a pretty good one. I don't know how we're going to come up with a more logical name than the one that you have. I distinctly remember the first time that I went through this, it just blew my mind thinking that I had to know the name of all of these structures, and I just had to memorize it all. It was such a relief when I realized that the names actually made some sense, and you could use them to scare people. Right? I had a lot of fun with scaring people. Believe me, I had a lot of fun going home during Thanksgiving break and just talking about fructose 6-phosphate and glucose 6-phosphate. No, that hexokinase, man. That's one of my favorite enzymes. It still to this day is one of my favorite enzymes. And so again, you can have some fun with this stuff because it shouldn't scare you the way that it used to scare me and it scared my parents. All right, so my parents thought that I was majoring in Latin as opposed to biology to talk about these things. All right, so glycolysis. How much longer is this going to take? It's a 10-step process. Good grief, we're only in the 2nd step. Don't worry, we'll go faster. I'll go faster through the rest. We start with this energy investment phase. We've invested some energy here, right? We got a little more energy to invest. How do you suppose that's going to happen? When you're talking about investing energy or extracting energy, at some point somebody's going to mention ATP. Okay? That's what happens when you're talking about energy investment or energy extraction in a biological context. ATP is going to be in there somewhere along the way. Look what happens. We get an enzyme that has two substrates. One of those substrates is fructose 6-phosphate, an old friend. We know that one from a ways back now. Another substrate is ATP. All right? The products of this chemical reaction are going to be ADP and a 6-carbon, I emphasize 6, but a 6-carbon sugar that now has 2 phosphates on it. So, a couple of things that I'm afraid I'm going to be insulting you because you don't need me to tell you, but this is an energy investment stage because we're taking ATP and we're pumping its energy into a carbon molecule, and we're ending up with ADP. That's energy investment. Later on, when we start talking about converting ADP to ATP, that's going to be an energy recovery or an energy recoupment process. Right now, we're still investing energy. And check out what catalyzes this reaction. Any great surprise that the catalyst, that enzyme, is going to have kinase as part of its name? I sure hope not. If it was, you dozed off during about 10 minutes ago. I was talking about what kinases are. Kinases involve, it's an enzyme that transfers a phosphate group from something to something else. Okay? So we've transferred a phosphate from ATP to fructose 6-phosphate. That's what kinases are all about. What about the phosphofructo part for the name? Well, what else did you want to call it? All right? We had fructose 6-phosphate. I guess you could have put phospho 6-fructokinase. I don't know, maybe you could say something about the 6. But when you come right down to it, where else was that one phosphate going to be? At the start of glycolysis, it's going to be on the 6th carbon, phosphofructokinase. And what do you make? You make something with, this is a good name, right? Fructose 1,6-bisphosphate. Okay? Fructose, not a big deal. That's what fructose looks like. That's what fructose looks like. 1,6, first carbon around the circle, 6th carbon around the circle. Fructose 1,6-bis, it's just a tip of the hat to the fact that there are 2 phosphates and phosphates because there's phosphates, so you got a phosphate at the 1 carbon and on the 6 carbon. If I were to quiz you and say which of these 2 structures has more energy, fructose 1,6-bisphosphate or glucose, intrinsic to its covalent bonds, I hope you're all going to bite on fructose 1,6-bisphosphate. Hope that's a no-brainer. In the context of what I've been talking about it is, but even if you just looked at those structures, same number of non-polar covalent bonds, but also a couple phosphate groups tacked on. Geez, that's just got to have more energy associated with it. Okay? If I were to ask you which of these 2, fructose 6-phosphate or fructose 1,6-biphosphate has, or bisphosphate has more energy associated with it, I hope you go with the fructose 1,6-bisphosphate because it's got 2 phosphates, whereas that had 1. Well, good news is we're through with the energy investment part of glycolysis. And we've got now a very energy-rich thing to play with, fructose 1,6-bisphosphate. It's ready to go. It's like the roller coaster ride, right? You started out at the platform. They took you up a little hill. Now we're going to go down, right? You could have gone straight down from the platform, but to make it a really fun ride, we juiced it up and you took you up from the platform, and we're about to start going down. First thing that's going to happen when you start to go down the hill, and we start now talking about extracting energy from glucose as opposed to investing energy in glucose, is we're going to break apart this 6-carbon molecule now into 2 3-carbon molecules, each with its own phosphate group. Each with its own phosphate group. I'm afraid I don't have a clever way to tell you about what's going on with this enzyme's name, aldolase. Okay? It's a one-off. There's no method that I'm aware of to the naming of aldolase, but aldolase's job is to break this 6-carbon bi- or bisphosphate molecule into 2 3-carbon molecules, each with their own phosphate. And then there's another enzyme that is so abundant and so central to metabolism that it gets the standalone name of isomerase. There's all sorts of other enzymes that are isomerases. There's only one isomerase. It's this one. And it interconverts between these 2 forms of the 3-carbon, 1 monophosphate molecule that comes out of what aldolase does. But again, we're done with the energy investment. Let's talk about what you can do with that 3-carbon molecule. So what's going to happen is we're going to take glyceraldehyde 3-phosphate. You make an equal amount of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate gets converted to glyceraldehyde 3-phosphate. And another enzyme called triose phosphate isomerase uses glyceraldehyde 3-phosphate and NAD+ as a substrate and creates NADH. Oh, it also uses inorganic phosphate. It creates NADH and 1,3-bisphosphoglycerate. Couple things. It's a shame that this gets straddled across 2 slides, but a couple things. Here's that 3rd class of enzymes that I was talking about. So we've got kinases, we've got isomerases, and we've got dehydrogenases. Unlike kinase, which you got to wonder what they were thinking to come up with kinase, right? Dehydrogenase makes a lot of sense in the name. Dehydrogen, right? A dehydrogenase takes a hydrogen off of something and it moves it somewhere else. Just as a kinase moves a phosphate group from one thing to another thing, a dehydrogenase moves a hydrogen from one thing to another thing. And almost always when you're talking about a dehydrogenase, one of the things that's involved is NAD or NADH. Here you get both. NAD as a substrate, NADH as a product. So dehydrogenases involve moving hydrogens from one thing to another thing, almost always in glycolysis, from NAD to NADH or NADH to NAD, one or the other. Sometimes it goes the other direction. Okay? Now, this NADH is NAD with a very high energy electron associated with it. Let me just tell you, NADH is a real dynamo. There's a lot of energy there. It's not useful for anything until you can convert it into ATP, but if you have the benefit of aerobic respiration and you can run your NADH through the electron transport chain, you can make some ATPs right there. 3, actually. Right? But that's separate from glycolysis, and that's only if oxygen is present. That's stuff that we'll talk about on Thursday. But look what we've done now, too. We've also been able to convert this energy-rich molecule into an energy-rich molecule that has 2 phosphates on it. That's got to be a good thing. That's got to be something that's going to help us make some ATPs out of ADPs in relatively short order. So what do you need to know? Well, dehydrogenase is a 3rd class of enzyme. Know that that means there's probably an NAD and an NADH involved. Triose, it's a 3-carbon sugar with 1 phosphate, ends up being 1,3, where is it on the 1st carbon and the 3rd carbon, bisphosphoglycerate. Glycerate is a 3-carbon molecule. So we had one 6-carbon molecule, and now we've made 2 3-carbon molecules. Both of these 3-carbon molecules have 2 phosphates on them, both the same, and we're ready to go to the races and make some ATPs. How are we going to make those ATPs? Well, there's two ways you make ATPs as cells. You do it through oxidative phosphorylation, which is code for the electron transport chain, which is something that we'll talk about on Thursday, or you do it through what's called substrate level phosphorylation. Okay? The way we're going to make some ATPs right now is substrate level phosphorylation. We've got an enzyme that has an active site that makes a nice comfortable spot for this phosphorylated molecule to sit, puts it in nice close proximity to a comfortable spot for ADP to sit, and it bends together in such a way that it smashes the phosphate into the ADP to make ATP and something else that doesn't fit as comfortably in the active site anymore. Simple idea. Substrate level phosphorylation, the way the enzyme is reducing the activation energy is it's helping bring together and hold steady something that's got a phosphate group next to something that has an ADP, and we're going to end up making ATP just because of this enzyme bringing those two players together. So, check out what goes on. 1,3-bisphosphoglycerate, 2 phosphates, ends up becoming 3-phosphoglycerate, 1 phosphate attached to that third carbon. And we've taken 2, because there were 2 of these 3 carbon molecules that run through glycolysis for each glucose. We take 2 ADP, and we've made now 2 ATP. This is what we're talking about. We're talking about wanting to make ATP. We have recouped our investment that we made at the beginning of the process. We've gotten 2 ATPs out. We had expended 2 ATPs. We got 2 ATPs out. Look at the name of the enzyme that catalyzes the reaction. It's a dehydrogenase, obviously. Oh, it's crazy. It's not dehydrogenase. Dehydrogenases involve NAD or NADH. They involve the transfer of hydrogens. Wrong. Right? Is it an isomerase? No. What else do we got? We got a third class of enzymes. It's a kinase. There's a phosphate group that got moved from one thing to another thing. We moved a phosphate onto ADP. That's what kinases do. Right? So no big deal with the name. That's a kinase. And it's phosphoglycerokinase because here the product of the reaction is phosphoglycerate. So no great mystery with the name there. But we've gotten some energy out of it. Let's keep going. Mutase and isomerase are equivalent. If you wanted, you could call this enzyme phosphoglyceroisomerase. A lot of people prefer mutase here. Right? What does that mean? That means we're going to convert something with a chemical structure into something else with the same, with a chemical formula. We're going to convert it from one thing with a chemical formula to another thing with the same chemical formula, just a different arrangement of bonds, right? It's an isomer. So we convert 3-phosphoglycerate to 2-phosphoglycerate. Long and the short of it is we move that phosphate from the third carbon to the second carbon to get things ready for the next step. The next step is an enolase. We're going to kick off a water molecule and make phosphoenolpyruvate. You might want to contemplate at your leisure sometime after class, is this got more energy or less energy than that? I'll tell you, it's a spontaneous reaction to go from here to here. That means this has got to be something that's got more energy associated with it than this did. But you know, the thing that you're kicking off doesn't have a lot of energy to begin with, and so there may not be that much difference in the energetics, but that might be something for you to consider. And then last but not least, the 10th step in this process. I told you a tale of 10 enzymes. The 10th and last enzyme is an enzyme called phosphoenolpyruvate, pyruvate kinase. It's got the word kinase as part of its name. That means we're transferring phosphates from something to another. This is an energy yielding step. That should mean that we've taken a phosphate off of something and tacked it onto ADP to give us ATP. There are 2 because there were 2 of these 3 carbon molecules that ran through right? We started with a 6-carbon molecule, and we end up with a 3-carbon molecule now, 2 of them for each glucose, but that 3-carbon molecule is called pyruvate. Couple big players. Glucose is a name that you should probably remember. Pyruvate, beginning of glycolysis, end of glycolysis. We've broken glucose into pyruvate. Okay? Along the way, we have made a total of 4 brand spanking new ATPs, shiny, crisp $1 bills, and we had to pay 2 crumpled up old $1 bills, 2 old ATPs to get this process started. All right, this is just zoomed in versions of all the things we've been talking about. Let me tell you that what we have next in order here is some talk about the citric acid cycle. The long and the short of the citric acid cycle is this. The citric acid cycle is how we milk that 3-carbon pyruvate for all it's worth to convert it into 3 1-carbon molecules carbon dioxide. Okay, so that's what's going to happen in the citric acid cycle. The way that that happens is a cycle, and we will talk about that on Thursday. This is it in its overview. It's going to be a deja vu sort of thing with the glycolysis. But before you go, let me leave you with that happy thought. Here's a question that I'll put on the next test for you, just to help you collect your thoughts about what we've been talking about here today. Other than that, though, I'm done. See you guys on Thursday, and we'll talk about the citric acid cycle and the electron transport chain.